Hello friends, welcome to Dr. Ossai Physiology Academy, DOPA for short. This is the place where we make the learning of physiology easy, exciting, and effective. Thank you for joining me. And if you're new to this channel, you're especially welcome. And if you love the content that we share, kindly click the like button and also the subscribe button. And do not forget to turn on the notifications so that you don't get to miss any new content that we drop. Let's grow the channel together. Thank you. So today we're going to be dealing with gastrointestinal hormones. Now, like we said, that two major ways the body communicates is through electrical. Electrical means and chemical means. Okay, electrical is through nerves. This is through chemical messengers. Okay, so there are so many chemical messengers that influence gastrointestinal function. We know about the electrical a lot. We've talked about the enteric nervous system. Okay, and also its influence. The influence of the autonomic nervous system, parasympathetic and sympathetic. So, but now there are many other chemical messengers that they are too numerous to mention. Okay, that's not what we're going to be doing today. We're going to be focusing on a special type of chemical messenger that are known as hormones. And hormones, they walk through the endocrine manner. You know that other messengers, they have the juxtacrine, neurocrine, paracrine, ways in which they communicate. But we are interested in endocrine, which is the cell communication at a distance. In the sense that the chemical substance, hormone in this case, is secreted into the bloodstream, circulates, and goes to the target tissue or organ or cell to influence a particular physiological response. So in this case, there are hormones that influence gastrointestinal functions. So how do we characterize these hormones? They have to fulfill certain criteria for us to really be sure that this is a gastrointestinal hormone. And one of them is this. It means that when the presence of food or whatever substance that can stimulate the GIT, when there is the presence of a stimulant, it influences a physiological response at another part, not just in the same part. You know, when it influences in the same part, it could be paracrine. You know, paracrine is nearby, just a short distance, travels through diffusion. But this one, it doesn't travel through diffusion. It has to go to the bloodstream and influence. So stimulus at one part of the GIT that is applied at one part of the GIT, stimulus applied at one part, influences GI function at another distant part. Another distant part of the GIT. Okay? That is that. Then number two is that, first of all, to be sure that it's really a chemical substance that is causing that physiological response at another part, you have to cut off the nervous supply to that part. So nervous supply is cut off. So after nervous supply has been cut off, for example, gastric acid secretion is influenced by acetylcholine, which is released from vagus nerve parasympathetic stimulation. So after you have cut off the vagus nerve, is there another chemical substance that is released that can also influence acid secretion. So you've cut off the supply, and that physiological response is still there. Then it shows that, yes, this thing is through a chemical means that is being influenced, is making it happen. Okay? So another one is that mucosa, GI, we're talking of gastrointestinal hormones. That means they're hormones that are, first of all, they are released from the gastrointestinal tract, they go to the blood and come back to affect the same gut. That's what it means. So it means that when you take out the extract, an extract of that gastrointestinal mucosa, you take it, and when applied and released into the blood, brings about that action. So these are just steps to ensure they fulfill, the hormones we are going to measure, they fulfill all these steps. Okay? So an extract of the GI mucosa, when injected into the blood, brings about the physiological response in a certain target cell in the GIT or target tissue in the GIT. That is another one. Then finally also, another fourth criteria is that this substance can be separated, identified specifically. Okay, it's not a mixture of different unknown different things. It is very specific. You can identify it, purify it, and synthesize it. It can be identified, purified, and because it can be identified and purified, it can also be synthesized. So these things, there are 4 major substances that fulfill all these criteria. And we are going to mention them. One of them is called Gastrin. Gastrin, number 1. Number 2, Cholecystokinin. Also known as CCK. Number 3, Secretin. Okay? Then number 4, it's known as GIP, but look at the name. It has a very long name. Glucose dependent insulinotropic, insulinotropic polypeptide, or simply peptide. Okay? So GIP. It has another special name. Okay? Let me write it here. So it's also known as gastric inhibitory peptide. Okay? The same GIP. So these 4 substances are the main gastrointestinal hormones that fulfill 1, 2, 3, 4. They're purely hormone. They are pure hormones. Okay? So now, what we're going to be doing in discussing these hormones, we're going to follow them systematically. Okay? So now, in discussing these hormones, first of all, we're going to talk about the class. The class of chemical substance. Okay? The number 2 that number two thing that we need to talk about in discussing this hormone is where is the site of production? Site of production. And another thing we should be interested in is what particular cells secrete it. Cells that secrete the hormone. So the next thing that we need to discuss when talking about this hormone is what is the stimulus for its release. The stimulus for its release. What stimulates it to be secreted? Okay? Then the last one is now the physiological action. Okay? So under these 5 things, we're going to discuss this systematically, one after the other. And it's always good to think about the different parts of the GIT. Does it influence this part? Does it influence that part? To help you not to miss anything. All right? So we're going to be doing that after this break. Welcome back. So we're going to be taking them one after the other, following these steps, these points you need to note. Very important points to note whenever you're talking about these hormones. So let's start with gastrin. Now, in fact, to make it easy, they are all of them. This chemical substance, all of them are peptides. So that one has already struck off for all of them. The class of chemical substance, these hormones, the 4 of them, they are all peptides. So we call them gastrointestinal peptides. Okay? So that is the same for all of them. Then the site of production, only gastrin is produced in the stomach, specifically the antrum. Antrum of stomach. Why these remaining 3? They are all produced in the small intestine. Okay? So those are the 2 major things that they apply broadly to all of them. They are all peptides, 3 of them, small intestine. Only gastrin is secreted from the stomach. So let's continue with gastrin. So what cells secrete gastrin? The G cells of the stomach are the ones that secrete gastrin. Now, important, the stimulus. What stimulates gastrin release? Now, very important to note. When food enters the stomach through distension and all of that, the presence of food, one of the things that stimulate, nervous mechanisms are always faster than chemical mechanisms. So immediately, the enteric nervous system is stimulated and the parasympathetic. So the parasympathetic nervous system being stimulated, what does it do? It leads to the secretion of acetylcholine. Okay? So acetylcholine now does what? It triggers the secretion of acid. 2 things. So immediately food enters the stomach. The first step triggers the parasympathetic that releases acetylcholine. Okay? So acetylcholine does what? It goes to stimulate some acid secretion, HCl secretion. And the next thing that acetylcholine does is to stimulate the release of gastrin. So acetylcholine stimulates the release of gastrin. But that's not all. This HCl being secreted is part of what activates pepsinogen. It turns it into what? Pepsin. Then what does pepsin do? It breaks down proteins into peptides and other forms, peptones. Then these peptides comes and stimulates gastrin. So the presence of peptides, but you know that when you take food, it's proteins you are taking, not peptides. So something has to break it. So that means this has to be secreted first, activating this, breaking it down before these peptides come. So the first thing is that acetylcholine stimulates gastrin and then stimulates HCl secretion, which further produces this peptide and potentiates this action of, that means it helps to multiply the effect of acetylcholine stimulating gastrin. So now a lot of gastrin is being secreted. Now the next thing is, what is the physiological action? Very straightforward. From the name gastrin, it leads to the secretion of gastric acid, which is HCl. Okay? So gastrin, major physiological function that you should never forget. Even if you forget any other thing, you should never forget it. It leads to the secretion of hydrochloric acid. Number one. Number two, it helps to stimulate gastric motility. Okay? Gastric motility, that mixing and grinding movement, and also gastric emptying. So generally, gastric motility stimulates gastric motility. But that's not all also. Gastrin has some other special effect. Now, if you notice when we're talking about gastrointestinal reflexes under gastrointestinal motility, okay? Just check the lecture. The lecture is there. You can revise it. So we said that there's something, a reflex known as gastroileal reflex and gastrocolic reflex. So these reflexes are brought about by the enteric nervous system. But gastrin also plays a role. So you see that both the nervous system, nerves and hormones and chemical substances, they help themselves. They play the same role. Okay? So number three is that it also, when gastrin is released from the stomach, it also joins to help to stimulate motility in the ileum. Ileal motility. You know, it's gastroileal, gastrocolic. The colic is talking about the large intestine, the colon. And the motility that happens in the colon is called mass movement. So it helps to bring about mass movement. It's part of what helps, okay, in conjunction with the enteric nervous system and the parasympathetic nervous system, all of that. So these are basically the functions of the hormone gastrin, released from the antron of the stomach by the G cells. All right? That's simple enough. Now let us enter the next one. Cholecystokinin, CCK for short. We've talked about this and this, okay? So now is the cells. What cells secrete it? Let's clean this. So cholecystokinin, they are secreted by the eye cells. The eye cells of the small intestine. So next, what is the stimulus for its release? Stimulus for its release is amino acids and fats, the presence of fat in the small intestine stimulates the release. Then next, you want to ask yourself, what is the physiological action? And remember we said during biliary secretion, also check the lecture, that what gave cholecystokinin its name, cholecystokinin, is the fact that it helps to contract the gallbladder. Cholecystokinin. Okay, so number one function of cholecystokinin, contraction of gallbladder. That's one of them. Then number two, contracts gallbladder. It also helps to stimulate the acinar cells of the pancreas to release pancreatic enzymes. You know, pancreas has the enzymatic part and also the bicarbonate, watery electrolyte part. So its major function is the enzymatic part, which is released from the acinar cell. You know, you still have the ductal cell. Okay? So, number two, stimulates acinar cells of pancreas. Acinar cells of the pancreas. To do what? To release pancreatic enzymes. To release enzymes. So, enzyme-rich pancreatic secretion. Cholecystokinin does that. Number three, cholecystokinin. It also helps in the relaxation of the sphincter of Oddi. Because now it has made this one contract. It has stimulated this one to, both of them join to now form the pancreatohepatic duct that enters the duodenum. So it will not be complete if that sphincter is blocked. So part of what it does too is to release it so that these things that it has stimulated will enter the duodenum. So it leads to relaxation of the sphincter of Oddi. All right? So it still has some other functions. And that has to do with the stomach itself. It inhibits gastric secretion and gastric motility. Number four, it inhibits gastric secretion and emptying, to be more specific. Okay? So these are main functions of cholecystokinin. Very straightforward. Okay? So let's talk about the next one, secretin. So site of production, small intestine. So now the cells that secrete secretin. Simple. S cells of the small intestine. Okay? So next is the stimulus for its release. The stimulus for its release, you will see why that is the stimulus. The stimulus for its release is acids. Acid in the duodenum, that's just a small intestine. The small intestine. And why is that? Now check the physiological action. The physiological action that is very prominent of secretin is that it is the main hormone necessary for the stimulation of bicarbonate. From where? Both from the pancreas and even from the bile ducts. You know, the bile ducts from the bile salts and all that they secrete. The ductal cells that contributes up to 50% of the volume of bile secretion. Secretin helps to also stimulate bicarbonate secretion from those ductal cells. So the same thing, but most of the bicarbonate comes from the pancreas, okay? Which is also ductal cells. You know, the pancreas has acinar cells, okay? But the ductal cells is where it stimulates. The acinar cell is cholecystokinin. So it stimulates, number one function is that it stimulates bicarbonate secretion, okay? Both from the bile ducts and pancreatic ducts. Number two is that it inhibits gastric secretion and gastric motility, just like its friend, cholecystokinin. They are friends. They have, because another thing I need to mention is that they potentiate the action of each other. This potentiate means they enhance and magnify their main action. You know, this one has a special action of stimulating the enzymatic part of pancreatic secretion, the acinar cell. So secretin helps it to do that work. Then cholecystokinin helps secretin in the stimulation of bicarbonate secretion. Okay? That's that they help each other. But this secretin does the main work. Cholecystokinin helps it to enhance its work. Okay? So it inhibits what? Gastric secretion, gastric acid secretion, and motility. Okay? So that's basically what it does. Basically, bicarbonate, both from pancreas and bile ducts, inhibits gastric secretion. So the next one we're going to deal with is GIP. Now, look at the name very well. Glucose dependent insulinotropic polypeptide. Now, the cells there is K cells. The cells that secreted K cells of the small intestine. So now, what is the physiological action? It just has one simple physiological action. And that is what? It enables, stimulates the secretion of insulin from the beta cells of the pancreas. That is all it does. All it does. Function. Stimulates insulin secretion from beta cells of pancreas. Okay? So we forgot to mention the stimulus. The stimulus for its release is all the different classes of food. That's something that is special about GIP. So both amino acids, glucose, or you can say carbohydrate, but more specifically glucose. Then fats. Okay? That's why it's called glucose dependent. So glucose, very important, because insulin has to do with glucose. Insulin has a relationship with glucose. Insulin helps glucose to enter into different cells of the body. So glucose stimulates it, amino acid, fats, all right, from the small intestine. So its job is to, and this, because of this, its job, it has a special name. It's called an incretin. Why? Because it is a hormone that helps the secretion of another hormone. Okay? Insulin is a hormone. Very important hormone when you are dealing with endocrine physiology. So it's an incretin. It helps to stimulate the release of another hormone. That is just all it does, GIP. So these are the main 4 gastrointestinal hormones. There's another 5th one. It's also a hormone, but not so much is known about it like that. It's called motilin. Motilin, secreted from the upper duodenum. Okay? So what it does is that there's something called MMC. MMC as migrating motor complex. What does it mean? Migrating motor complex. It's also called migrating myoelectric complex. So what it does is that during fasting states, when you're fasting, you're not eating for a long time, it helps to stimulate motility in the gastrointestinal tract. From the name itself, motility. So it helps gastrointestinal motility in the time of fasting. Okay? So we call it interdigestive migrating motor complex. Okay? So that's what it does. So these are the 4 main hormones you must know about. And once you have these things behind your back of your mind, this one, all of them are peptides. This only gastrin is from the stomach. The rest are from the intestine. Then the cells that secrete it. Very simple to know. They are all in numbers. One is G cells, I cells, K cells. Okay? Then S cells. G, I, S, K. G, I, S, K. GISK. Okay? So the physiological action just is simple. Gastrin, gastric acid, cholecystokinin, contraction of gallbladder, enzyme, and so on. Very easy stuff. So this is basically what you need to know about gastrointestinal hormones. Right? So for further reading, I've written a book on gastrointestinal physiology. It's called Gastrointestinal Physiology, broken down for you. Very easy to read, easy to understand textbook. And also other books in physiology in 7 other areas. Okay? 7 books in physiology I've also written. The links are there in the description box. So just click the link you see. It directs you to Kada Books where you can download the soft copy just for a token. So to help you and get a little more details about these things. Also, in the description box is the link to our website. You can visit it too, so you can get to know more things we do to help and make the learning of physiology easy, exciting, and effective. See you in the next video.